So today we move on to the female reproductive system. So we talked about the male reproductive system, and we're going to talk about the female reproductive system, but part of that is going to be talking about how they differ, how they're the same, and looking at the anatomy and the comparisons and the development. So of course the female reproductive system has two sort of main functions. One is producing the eggs in the ovaries, and the other is incubating the fetus during pregnancy. So we'll be talking mostly today about sort of the general process, the egg production, and then next week in our last class session is when we'll actually talk about pregnancy and embryonic development. So here's our female reproductive system. But I want to talk about where it comes from. So in the early embryo, we have this indeterminate state. And this indeterminate state has the early gamete, the protogonad, where the gametes are going to develop. And this right now can develop either into a testis or an ovary. So you have this protogonad has the ability to develop either way. Now we have separate ducts here, the Wolffian duct in here, and the Mullerian duct here. So the Mullerian duct in a female will develop into the fallopian tube. The Wolffian duct in a male will develop into the ductus deferens. So those tubes differ, but all of the other structures, so here we've got a urogenital sinus, here we've got these protogonads, all the other structures are going to start off the same and develop from there. You'll see some very early differentiation in these gonads, but this is sort of this precursor state. And then we have the activity of the SRY gene, and the SRY gene is going to send the fetus down either the male development path or the female development path, and you either get this development of the testis and the ductus deferens, or you get the development of the ovary and the Mullerian duct. Now on the outside, we're going to see something very similar. So the initial state, you have this genital tubercle at the tip here with the urethral folds, the cloacal membrane in the center, and the labioscrotal swelling. Here, this is very early on, you're seeing this little tail. In most vertebrates, that would develop into a full tail. In us, of course, that tail goes away. It's reabsorbed back into the body during development. And so this is the same basic set of structures, regardless of what whether this embryo eventually develops into male or female. And so we can look here at the genital tubercle, which is the glans, the phallus structure here, and then the urethral fold in the center, the labioscrotal swelling, the urogenital membrane in the center. So as these develop, the glans is going to develop into the glans penis in the male or the glans clitoris in the female. So either way, that becomes the glans there. And so down at the end here, you'll see the glans clitoris here in the female, the glans penis in the male. The rest of the penis structures will develop here in the penis and into the internal clitoris in the female that you've seen in lab before. We'll talk about more. And then you have the urethral fold, which in the male is going to close up and form around the penis here, and in the female will remain open and form the labia minora the inner labia surrounding this. Then we have the labioscrotal swelling. So the labioscrotal swelling in the male will then form the scrotum and close up over this base. And then you have the scrotal raphe, which is the line down the middle of that closure. Now, it is a fairly common developmental issue for this not to close all the way, which will allow for an opening somewhere other than this final urethral point, because this is a fairly late closure. This closure happens later in the fetal development. You can see here, you're still seeing this opening. And of course, in the female, it will remain open, and you'll have the vaginal orifice here with the urethral opening down here in between the labia minora, whereas in the male, it's up here at the end, past that closing on the glans penis. So what you can see here is that these two develop from the same basic structures, and that means that there's actually a wide range in between. There are a lot of people who fall somewhere in between these two because the development didn't wind up all the way at one end or the other. And we're looking at the case study, of course, which covers one particular example of someone who's in an in-between point. So now that we have sort of talked about the development, I want to focus in on looking at the external female genitalia, and we'll continue on to the internal female genitalia. So the external female genitalia, you have the mons pubis, which is a padded area. There's a fat pad here for protection. There's coming in, we've got the labia majora, forming the outer lips, the outer protective area. Coming in from there, we have the labia minora. And at the tip of the labia minora, we have this glans clitoris. Inside the labia minora, we have the urethral opening, and we have the vaginal entrance. You're seeing here the remnant of the hymen. Now, the hymen is never a complete covering, and this can become torn through a lot of ways, obviously most famously through sexual intercourse, but a lot of people have the hymen torn before that point. So this is the external genitalia here, and then down here we have the perineum and the anus. Now, this is an example of intersex genitalia. As I said, there's a wide range in between. Just because we tend to think in male and female doesn't mean everyone actually falls neatly into those categories. And when you look at someone's genitalia, it doesn't necessarily automatically tell you a lot about their genetics. Obviously, most people who have female external genitalia are going to have XX. Most people who have male are going to have XY, but there are exceptions both directions. And so then we can move into the female reproductive system. And we're going to work through the anatomy of this sort of as we work through the functions. But here's sort of the overview, and I'm going to end this one here.